Welcome to the deep dive. Today, we're looking at some incredible research that really forces us to revisit one of the oldest arguments in philosophy. And it's an argument that has kind of become the central crisis for modern AI.
Yeah, you're talking about nature versus nurture, but, on a philosophical scale.
Exactly. You've got John Locke on one side.
Tabula rasa,
Yeah.
the blank slate,
hmm
the idea that the mind just starts empty. It's waiting for experience to write on it.
And then on the other side, you have someone like Immanuel Kant who argued, no, that's impossible. We have to have some kind of innate structure already there, a priori categories, just to make sense of anything.
And if you look at how we build AI, deep learning today, it has overwhelmingly gone for the blank slate model.
Oh, completely. You boot up an artificial neural network, and what are the weights?
They're random, just Gaussian noise, basically. It's a field of, computational static. It's designed to be a blank slate.
And then we just throw astronomical amounts of data at it,
Yeah.
petabytes. And we hope that it carves out some meaning from that void. It's brute force.
But what if that's not how biology does it?
Yes.
What if the reason biological intelligence is so efficient is because it completely bypasses the blank slate problem?
And this is where the research we've been digging into, specifically from Dr. Tal Sharf's lab at UCSC, just delivers a massive shock to that whole paradigm. Their findings are suggesting that the human brain is, well, it's decidedly not a blank slate. It actually boots up with a pre-installed OS already running.
And that's the inflection point for AGI.
Huh.
So that's our mission for this deep dive, to get into the biology, this discovery of protosequences, and figure out what it means for building real intelligence.
Okay, let's start there with the biology, because for the longest time, understanding early brain development was, well, it was almost a black box.
Right. You can't exactly study fetal brains and the alternative growing cells in a flat petri dish. It's just too simple. It's not how a brain is structured.
It doesn't have the architecture.
Exactly. And that's what's so powerful about the organoid revolution.
Yeah.
We can now grow these tiny self-assembling 3D spheres of brain tissue.
From human stem cells, right, the iPSCs.
Yep. And you get something that starts to form layers. It builds complexity. It mimics a real developing cortex. It's the closest we've ever gotten to watching the process unfold.
But having the structure is one thing. You have to be able to, listen to what it's doing.
Precisely. And the listening equipment the Sharf lab used is frankly what made this whole thing possible. They're using these COS-based microelectrode arrays, MEAs.
The analogy I read was pretty compelling. Is like instead of having one microphone in a giant, noisy crowd, you're placing thousands of tiny individual microphones right inside the crowd. You can hear individual conversations.
That's the key. High density. It lets them triangulate the electrical signals from single neurons deep inside this 3D tissue. Yeah. And critically, they can do it non-invasively.
So they can just watch it over time.
For weeks, months even. That's the longitudinal monitoring part. And that's how they found the protosequences.
Right. So what did they see? It wasn't just random firing.
Not at all. As the organoids got more mature, the neurons started to organize themselves. Their firing formed these coherent, structured patterns over time.
And this is with no input, right?
Zero sensory input. No light, no sound, no touch. The brain was just generating its own internal structure, completely on its own.
Sharf calls it a primordial version of the brain's operating system.
It's the perfect term. An OS sets the rules, right? The syntax for how to process information. And what they're seeing is the brain rehearsing those neural algorithms before it gets any data to process.
The efficiency of that is just staggering. It's not waiting to learn. It's pre-writing the template for learning.
And the way it's firing tells you it's optimized for exactly that.
This is where that idea of near critical dynamics comes in, which is a concept from complexity science. Can you unpack that a little?
Sure. So think of it like a tightrope. If a system is too ordered, like a crystal, it can't change or adapt.
It's rigid.
Right. And if it's total chaos, like pure static, it can't hold on to information. Near criticality is that perfect sweet spot right on the edge between order and chaos.
Okay.
And that's the state that's associated with the best possible information processing. And the brain just self-organizes into that state right out of the box.
And it wasn't just some uniform, neural juice. They actually compared different types of brain organoids.
That's right. They compared dorsal forebrain organoids, which become sensory and motor areas, with ventral forebrain organoids, which form deeper structures.
And they behave differently.
Very differently. The ventral forebrain ones formed these modular, localized clusters of activity much faster. The dorsal ones were more broad, more synchronous. It means the hardware is already being specialized before any data ever arrives.
So if you connect that finding to the bigger picture of the brain, it really makes you rethink what all that background noise in our heads is actually for.
Absolutely. This intrinsic activity, these protosequences, they seem to be directly linked to what we call the default mode network, the DMN.
That's the network that kicks in when your mind wanders, right? When you're not focused on a task, you're thinking about the past or planning for the future.
It's the predictive part of the brain. And if these organoids are showing DMN-like patterns without ever having experienced a world to predict,
It means the DMN isn't something that develops late. It could be the foundation.
it might be the root directory of the entire system. The default state of the brain is already a functional predictive machine.
This just flips so much of neuroscience on its head. Spontaneous firing used to be dismissed as, what, metabolic dark energy or something?
It's noise, but now it looks like it's the critical initialization signal.
Hmm.
It's the network building its own world model from the inside out.
It reminds me of those retinal waves in
A
development.
perfect parallel. Before an animal's eyes even open, waves of activity are washing over the visual cortex, pre-training it to see edges in motion.
The brain is generating its own synthetic data,
Exactly, to get the hardware ready for maximum efficiency.
which connects to sleep, right? If this intrinsic activity is the default, then maybe the awake state is just a modulation of that underlying dream state.
It flips the script entirely.
Yeah.
The dream state, the intrinsic state, becomes the primary one, constantly running diagnostics and rehearsals.
And that has huge clinical implications.
Huge. If the brain boots up with an OS, then neurodevelopmental disorders like autism or schizophrenia, they might not be runtime errors.
That could be boot error.
A defect in the self-assembly process, a corruption in those first protosequences that leads to a flawed default mode from the very beginning.
And with this organoid system, they can actually test therapies to try and restore neuronal signaling at that foundational level. It's incredible.
Now let's pivot this over to AI, because this biological fact just lands like a bomb in the middle of the current deep learning paradigm.
You're thinking of Rich Sutton's The Bitter Lesson.
Exactly. The idea that all you need is more computation and a blank slate, a low inductive bias, and that will always beat human-engineered priors.
But this research is the ultimate rebuttal. Evolution did the exact opposite.
Evolution invested heavily in a very high inductive bias initialization. It built complex, pre-wired structures to get insane data efficiency. The brain isn't a general-purpose computer. It's a purpose-built machine.
The tech community seems to get this, at least intuitively. You see it in the discussions around this research.
I love the analogies they come up with.
Like the Labrador puppy. It falls in the water for the first time and just swims perfectly.
It doesn't have to learn. The motor program for swim is like a pre-compiled binary. The water is just the trigger to run the code.
And the protosequences are the neural code for things like vision or causality, already there before the eyes even open.
It puts human learning in a totally different category. The classic one is the Dalmatian argument.
Right. A kid sees one Dalmatian and they get the concept for life.
Meanwhile, a model needs thousands, maybe millions of examples. Why? Because the kid has this pre-existing scaffold to hang that new information on,
Yeah.
the protosequences.
So this gets to the core problem. With our ANNs, we spend ages designing the architecture, the layers, the connections.
But then we just randomize the weights.
Yeah.
And what Sharf's lab shows is that biology provides both. It provides structured architecture and structured weights. It doesn't start with noise. It starts with a strong, functional blueprint.
So we're building these super powerful engines, but we're making them spend 99% of their energy just trying to bootstrap their own operating system from a flood of data.
We have to talk about how this affects the AGI roadmap, too, especially the idea of world models.
Like what Jan LeCun proposes, the idea that an AI needs an internal simulation of the world to make predictions.
Right, and models try to learn that by just watching tons and tons of video.
But the organoids,
The organoids show this incredibly structured, time-based activity without ever seeing a single frame of video. It suggests the protosequences are the precondition for learning a world model efficiently.
so the intrinsic activity is like the physics engine booting up first.
Yes, it's establishing the neural rules of time and space and causality before the physics simulation, with cars and balls and faces, ever begins.
Which points us toward a new path, developmental AI.
Exactly. Systems that don't just learn from data, but systems that grow based on some intrinsic pre-data dynamic. just like biology.
So what does that mean, practically? We need to completely rethink initialization. Things like Xavier or Kaming initialization.
They're just variance balancers. They stop the signal from exploding or vanishing, which is important, but they do nothing to structure the function of the weights.
So we need something like generative initialization.
I think that's the term for it. Imagine instead of random numbers, you have a tiny algorithm, a genotype, that runs for a bit.
And it generates the starting weights.
It generates a complex, structured weight matrix, the phenotype, that already has these functional oscillations, these synthetic protosequences, before a single piece of training data comes in.
The network would already be humming, rehearsing the syntax of reality. That's a totally different starting point.
And then, of course, there's the really radical idea, organoid intelligence, OI.
Just use the biological hardware itself.
Why not? If we can map the inputs and outputs of this system, why not use the organoids as, biological processing units, BPUs?
Well, the energy efficiency is the killer app there. A human brain runs on, what, 20 watts?
Try doing what these protosequences do in silicon. You'd need orders of magnitude more power. So if we can harness the biology, we solve the energy crisis of AI.
And finally, we have to actually implement dreaming, not just as some maintenance cycle.
No, it has to be the core driver of the structure. You have to give the system offline time to consolidate, to reorganize.
To process its own internal data.
Exactly. Mix real data with generative noise to build robust features. That's what sleep does. It's the OS running its own continuous self-correction.
Okay, so let's try to sum up the key takeaways from this deep dive. Biology, it seems, pre-computes reality.
The brain uses a genetically compressed operating system made of these functional protosequences. And that intrinsic activity, it's not noise, it is the critical functional signal.
Which means the strategy for AGI has to shift. It can't just be about big data anymore.
We have to start investing in the big prior, in what you could call model embryology.
The path forward isn't just feeding more of the internet to a blank transformer. It's about answering that old philosophical question. And Dr. Sharf's organoids seem to have given us a pretty definitive biological no.
Which leaves you with the final thought to really chew on. The big leap might not come when we finally feed an AI enough data. It'll come when we engineer a synthetic organoid, a digital network that's born humming with its own internal life, dreaming of the world before it ever opens its digital eyes.